Hey there! Today we are going to talk about scientific thinking. What is it? How is it different from other kinds of thinking? Why is it important? All that kind of thing. We're going to dive right in. So today, the goals while you're looking at this lecture are going to be such that you can list and describe what the scientific method is, including the steps within that scientific method. You'll learn to create a hypothesis, and there are two different types, and we'll get into that. But a hypothesis is always going to be stated as an if-then statement. You're going to learn to identify and define dependent and independent variables and what controls are necessary within an experiment. And then you're going to learn to form conclusions from a data set. And kind of maybe one of the most important things is to determine if an experiment is even valid. And hopefully also, I'll just throw this in there too, we also want to be able to determine if a conclusion that is stated is valid based on the experiment. So that's what we're going to get into today. So we hear these terms often a lot: critical thinking, scientific thinking. Right? Everybody wants to be a good critical thinker, and as educators, we're always pushing that. We want you to analyze what you hear and basically compare it to information that you know is true. And so is this new information true or not? All that is super important, but let's kind of define our terms so we know what we're talking about as we get into today's lecture. So critical thinking is the ability to evaluate multiple claims, which are defensible, all of them, and decide which one is more defensible. Scientific thinking is the capacity to evaluate the weight of the evidence that you see in favor or against a given idea. So science isn't interested in if things are equally defensible. Scientific thinking is usually aimed at providing an explanation of natural phenomenon, things that you see in nature, and have it be supported by an overwhelming amount of evidence, basically supported by all kinds of facts that gives you an account of what exists and how it functions. So if you have in science, if you have more than one position that is equally defensible with the data, so in other words, you could come to one conclusion or another given the data, then that is considered inconclusive. So the mechanisms can't be adequately identified. And so when we're thinking about just critical thinking, that's more of a broad skill of evaluating information and arguments from various sources to come up with some reasonable judgment. But scientific thinking says it either is true or it's not. There is no in between, and that's based on lots and lots of repeatable experiments. It's true every time. The evidence shows X. Okay, so that's how they are a little bit different from one another. Both are extremely important. I want each one of you to be able to think critically and to think scientifically, and that's kind of our goal. So I mentioned that at the end of a scientific experiment, you either have a solid yes or a solid no. So it may surprise you if I also say science explanations change over time. Well, how can those two things be held in the same hand, right? So if we do an experiment and our answer comes out yes, it may be that further in time, our body of knowledge or our new technologies allow us to come back to the same question with a different approach. And then our answer may be, oh, actually, no. And so it may be that our experimental mechanisms previously were incorrect, or there were assumptions when we made that experiment that were incorrect as well. As much as we try to correct for those using controls, which we'll talk about in a minute, there are still assumptions. right? So science is always based on evidence, and evidence can sometimes change. So that's just something to keep in mind as we're talking about scientific thinking and as we're talking about experiments. I will say sometimes right now in our society, we have a tendency to think, oh, but scientists are always right because these are facts. And scientists, one, are fallible just like every other human. And two, science explanations do change over time. And we'll get into later theories versus law, and we'll see how that plays into that. But just keep in mind, it is usually very fluid. And the most exciting thing to me about science is that there's always stuff we don't know. Always. There's always more to learn. And so that means we can always design a new experiment, always learn something new. So that is super exciting to me. One of the prime examples of things changing over time and science explanation is the atomic model. So it used to be earlier models, like in the 1800s, atoms were thought to be indivisible particles, right? They were the smallest things that made up all sorts of matter. And then later, things kept changing. Those scientific explanations, as more information came, started to become more complex, and we started realizing, oh, atoms are made up of electrons and protons, and then how do those work, and what does that look like? And so our modern model of atoms is our quantum model based on quantum mechanics, and that is what you guys are going to be familiar with atoms having a nucleus of protons and neutrons, and that's surrounded by a cloud of electrons that have a specific level of energy, and those stay within orbits. And so all of that changed between the 1800s and the 1900s. So again, new evidence, new technologies allowing our model to change. So if you had asked somebody in the 1800s, is there anything smaller than an atom? They would have said no. Factually, no. Based on all the evidence, based on all the experiments we've done, atoms are the smallest particles known, right? And that was well accepted at the time. Move forward into the 1900s, and that's no longer true. So even our knowledge base of what we would have thought of as scientific fact is constantly changing. And I think as a society, we often tend to think, oh, but we're so much more enlightened now. And we fail to see that somewhere our models currently are going to be fallible, and they will be replaced in the future as more information comes to be. And that's not always going to be true, but we do have to hold loosely to the knowledge base that we have and understand that more information will be coming. Eventually, I'll probably tell you this, but showing my age a bit, when I went to graduate school was when the gene banks were starting to be formed. Prior to that, we didn't have that information. We didn't have the genetic technologies that we obviously have now. And so during my graduate school time, they sequenced the entire DNA of this tiny little plant called Arabidopsis. It's basically a weed, but it's like the model plant genetic, the plant genetic model, because they had everything sequenced. Well, that was huge at the time. Like, oh my gosh, we know every single sequence that they have in its genome, even though it's a really small genome. And the whole DNA database didn't exist. It was being built at that time. And so, yes, I know I'm old, but I'm not that old. So things happen very quickly. New information comes in. And so we just have to be open-minded and use our critical thinking and our scientific thinking to realize that things are going to change over time. So how do we do our best to think scientifically, to start seeing our world in that way? The best way to do that is the scientific method. And this was really started to be developed by the Greeks. And over time, of course, got more and more specific. And so currently, these are the steps that we have within the scientific method. It always starts with an observation. which leads to a question. And then once you have your question formed, then you start to talk about your hypothesis. Hypothesis is basically, what do I think will happen? Or sometimes it's a why question. Why do I think this is happening? And then we design an experiment to test that hypothesis. And then we look at our data, which is number 5, analyze, and then we make conclusions. Now, each one of these steps is very specific. So by being specific and systematic in the way that we look at something, that's a very scientific way of approaching the world and how we think. So our observation step, this is basically just using all your senses, your smell, your sight, your touch, your hearing, your taste. And so if we're looking at this picture, we're seeing all these boats. We see these beautiful colored trees. And so those are just sights, right? We might be hearing lots of motorboats, or maybe not. Maybe these are all man-powered somehow. We may hear the wind rustling through the trees. Hopefully we're not going to taste too much out there, but in a different scenario, you might be. And so once you start observing, sometimes you will start to form a question based on those observations. So scientists generally are very curious people. They want to know why. They want to know, is that going to do that again? Why did that happen? I wonder what would happen if I changed this scenario. So it's a lot of curiosity being turned into a question. Okay, once we have our question, then we need to form an answer to that question, basically, that can be tested. So that is called a hypothesis, and we always write it in the form of an if-then statement. And the hypothesis needs to be able to explain what you expect to happen. So your prediction, basically. You need to be super clear on what it is you're saying is going to happen. But not only that, it has to be testable and measurable. So the reason it needs to be testable and measurable is if it's too vague, then you're never going to get your question actually answered. And within that, it needs to contain an independent variable and a dependent variable. And we'll talk about that in just a minute. So there are two types of hypotheses that we're going to write. The first one is what we call a null hypothesis. That term null just means no, not true, didn't happen, okay? And so basically we write our if-then statement as if nothing has happened. Nothing is what you predicted. So in other words, there's no relationship between the variables that you're trying to test. So in our examples here, we're saying the cat shows no preference for food based on shape. Doesn't matter what we throw at it, it's not going to have any preference. Or in the second scenario, notice that they have a yellow light and a red light. Those are some variables. And we're saying the plant growth is not affected by light color. Or the last one, we have a young woman and an old woman who are trying to play the violin. And we're saying age has no effect on musical ability. That is our null hypothesis. Notice I don't have them in if-then statements at the moment. Basically, the one on the left with the cat, I would say, if given different shapes of food, cats show no preference for food based on shape. Or the second one, if grown in different light color conditions, plant growth is not affected at all. No difference is seen. Okay, so that is what we're looking for, but basically a null hypothesis says there's no effect on changing a variable. So if we have a null hypothesis, then we always have to have an alternative hypothesis. And just to point out real quickly, we denote a null hypothesis by saying H0, hypothesis 0. And then your alternative hypothesis, we denote that by saying H1. Okay? So in this particular example, we're going to add some biofertilizer to our plants. And then we're going to ask it, does that affect growth or does it not? Well, as we saw in our previous slide, that null hypothesis would always predict that there's no statistically significant difference between our biofertilizer and our non-biofertilizer. So our null hypothesis is going to say, if I applied biofertilizer, I expect no change or no difference in growth. Okay? And then our alternative hypothesis, there is going to be a significant relationship between the variables. So in my alternative hypothesis, I'm going to say, if I apply biofertilizer, there will be a significant increase in plant growth. Okay, so do you see the difference between our null hypothesis and our alternative hypothesis? The way I like to think about this, often just the way I see it, let's say let me put this in red. Let's say we have our question here that we've come up with, and then based on that question, I should have two answers. I can either have my null hypothesis or I can have my alternative hypothesis. And once I do the experiment, then I'm going to be able to X out one or the other and say, oh, this particular hypothesis was correct, but the other one was not. Okay, so it's kind of like a flow chart, and I'm giving myself two alternatives for answering the question. So we have to design an experiment that is going to be able to answer our question based on our hypothesis. So basically, how do I design a controlled experiment? How do I make sure that I'm answering the question I want to ask? So here are a couple of definitions to get us started. The first is our independent variable. You know what independent means. It means it doesn't depend on anything, right? So in other words, in the experiment, it's the variable that I choose to change because I'm the one testing it, my independent part. So this is like the if part of your hypothesis. So if we were talking about that biofertilizer, it says, if I add this biofertilizer, that's my independent variable. Then there's our dependent variable. That variable very much depends on what the independent variable does. And so that's the one that I'm measuring. It's the one that I'm looking at to see if there are any changes. It's the response part of the if. So it is the then part of our hypothesis. So if I had started my hypothesis with, if I added biofertilizer, the independent variable, then I will see significant growth, the dependent variable. So plant growth in that instance would be my dependent variable. If my hypothesis, in this case my alternative hypothesis, is correct, then my growth will be dependent on my biofertilizer. If I add more biofertilizer, I'm potentially expecting that I will have more growth. Does that make sense? Okay, so then what I also need to do is look at the controls. So I have to keep everything else in the experiment fair or the same. Otherwise, I won't know if I'm actually testing the biofertilizer or if I'm testing something else. Can you imagine if I added fertilizer, but I only watered one plant and not the other? Then I would have no idea if my growth was affected by the biofertilizer or the amount of water that I put in there. So our control variables are things in our example, like the amount of water has to be exactly the same for both plants. The amount of light that they get and the amount of hours they're exposed to it have to be exactly the same. The amount of humidity in the air that it's growing in also has to be exactly the same. The soil has to be exactly the same. Okay, so we are controlling as much as we possibly can in order to only vary our independent variable. Now, some of that, we're also going to have to make some assumptions, and that is the tricky part about designing an experiment. So even if I use the same exact seeds from the exact same kind of plant, there is going to be a slight variation in how one grows versus another, just straight out of genetics, right? But what I can do to help that is plant as many seeds as possible so that that variance in the natural growth kind of averages out. Okay, so that's another thing to keep in mind, that sometimes we have to just go ahead and understand that there are assumptions that may not be able to be controlled for. But to the best of our abilities, we are going to provide controls that keep the experiment fair and the same for all of the plants in this particular case. Okay, so we have planted these plants, we've added biofertilizer to half and not to the others. Everything else we could possibly keep the same, we have kept the same. Now we need to collect our data. Okay, so this is an interesting part also. So just something to keep in mind. Let's say we're measuring height. That's what we're going to count on. Plant height is how we're going to measure whether or not the plant growth was affected by the biofertilizer. Well, that's one way to think about it. We could also take the plant and weigh it. So that could be a way of measuring it. All kinds of options, okay? But for our simplicity, we're just going to say we are looking at the height variance between those that had biofertilizer and those that did not. Okay. One of the things that scientists will calculate, and we're not going to go into details today, but I just wanted you to know that it's out there so that you know that it's something to consider. And that is what's called the p-value. The p-value is the probability that our results happen just by chance. Not because we added the biofertilizer, not because we kept everything else the same, but just by chance. So a low p-value, in other words, it's not really likely that this is just going to happen by chance. That means we can have pretty high confidence in the results that we get. Okay? And so once we have collected all of our data, we're going to analyze it. Here, let me just tell you the way I do things because I'm not great at statistics. I will take my data over to the statistics department and say, can you please analyze this and tell me if my height from my biofertilizer plants is significantly greater than the ones that did not receive biofertilizer. Okay, it's great to have other people to collaborate with who have strengths that you do not have. So we've collected our data. We have found out, oh my gosh, they're so much bigger when we add biofertilizer. And so in that case, in that situation, then we are going to accept our alternative hypothesis and reject the null hypothesis. Because the null hypothesis would have said that there is no significant difference between the two groups. And our alternative hypothesis is saying, yes, there is. Okay? And so that's really important. That tells us, hey, this biofertilizer might actually do something. Okay? And the great thing I feel like is once you get to either one of these, it always leads to another question. Okay? So if I said, yes, my plants with biofertilizer grew so much more, well, now I might want to know why, right? So maybe I will break down that fertilizer into its parts and maybe do the same experiment, but with only parts of the fertilizer to see which one has the significant effect. So there's, like I said, there's always more to learn, which is great. But our conclusion then is basically the acceptance of the alternative hypothesis. We could say when biofertilizer is applied to specific plants in a specific situation, then there is a significant increase in growth as measured by height. Like you have to be very, very specific about what your conclusion is because it can't be general, because if it's general, then people can take the wrong assumptions from it. They could say, if I apply biofertilizer to my corn, and let's say we didn't test corn, then I'm going to have so much more growth. And in their head, they might think if I apply this biofertilizer, I might have so many more corn stalks. Well, that's not at all what we tested, right? So our conclusion has to be very specific about the outcome and what exactly we tested. I hope that's making sense. Okay, so when we are reporting our data, I kind of wrote this a little more succinctly so that your conclusion let me try it again. Your conclusion, like I said, should be very clear and very specific. It was predicted that when we apply biofertilizer to Arabidopsis plants, you might even put in there at such and such percentage, then significant growth is shown via height. Okay, just coming up with that off the top of my head. And then you would say, this was affirmed by our results. The results showed significantly taller plants in those that were treated with biofertilizer than in those that were not. Now, this next part says, then state your reasoning for why this happened. You may or may not put that in there. Oftentimes, I don't put in the reasoning why. And my reason for that is because we didn't test why, right? So my personal thought is that should never be included in your conclusion, actually. So I'm going to scratch that right there. You can think about that. Oh, I'm not doing that very well. Here we go. Okay, so I don't want you to state your reasoning for why it happened. You just state that it happened. Why might come later, right? And then we need to present our data usually in some picture way, in a graph, in a, that sort of thing, so that people have a visual. And it's extremely important that the data be able to stand on their own within that graph. A person should be able to look at your graph or pictogram and be able to tell what the experiment concluded without even reading your whole report. Okay, so that's going to be significant. Here we have a sample experiment where somebody looked at the temperature at various latitudes. And I'm going to just have you pause here and look at observations, your questions. What hypotheses would you come up with? What would be your null hypothesis? What would be your alternative hypothesis? And when you look at the experiment, what's the independent variable? What's the dependent variable? What controls would you need? And maybe think about what assumptions are made. Okay. One thing I do want to say, I just realized this, when you do a graph, the dependent variable, the thing that depends on what we manipulate, is always on the y-axis. That's where we plot that. And then the independent variable, I'm going to do that in green. This is what we change. In this case, the latitude is always on the x-axis. That is always the case. So if I were to give you a graph, you should be able to look at it and say, oh, the temperature is definitely the dependent variable because it's on the y-axis. Or, oh, the latitude is definitely the independent variable because it's on the x-axis. Okay, so that should always be obvious in any graph that is produced. Okay, so we use the term data just in everyday language, kind of thrown around quite a bit. So I just wanted to kind of talk real quickly about quantitative data versus qualitative data. So quantitative data, that's things that you can actually measure. Just think this is numbers, right? This is how much does something weigh, how long is it, what's the temperature, something that you can put into numbers. Qualitative data is based on your observations. So in other words, well, here, I'll give you an example. A qualitative data is I could say, this music is loud. Okay, that's a qualitative statement. The quantitative statement would say, this music is at 100 decibels, right? That's a number. That was actually a measurable number. That's a quantitative data. Okay, or I might say, I don't know, this object is red. Okay, that's qualitative. Quantitative would say that reflects back, I can't even remember what red light is, let's say 400 nanometers, okay? They reflect back that wavelength. So there is a difference between the two. You can graph quantitative data. Qualitative data, that's a little trickier. Okay, and here's the part that does get me going if you want me to be on a soapbox at some point. That is objective versus subjective. And in science experiments, there's no room for subjectivity. It is all objective. And so you can think of objective being non-biased. Okay, so it's a fact, it's measurable, it's a number. It's not colored by your personal feelings. When something is subjective, which is perfectly fine, it doesn't belong in science experiments, but it's biased, it's colored by your feelings, it's your opinion, it's what you write when you write an opinion piece for a letter. I'm sorry, for a newsletter. If you are writing a scientific article, it should be objective. It should be about math, it should be just experimental. Honestly, the way I put it is it should be very dry. It should feel very boring because it's just full of facts. It's not colored with flowery language, none of that. Okay, so that is a difference. And the reason I say that is because so many times there are, quote, science articles written for public consumption, and it's completely tainted with subjectivity and bias. It's not just about the facts that allow you to come to the conclusion. So I want you to be hyper aware of that. It's notoriously all throughout the environmental movement. You hardly ever get a scientific article about anything environmental that isn't sensationalized because it's picked up by more popular and secular magazines, podcasts, whatever. So you do have to be aware of those differences, recognize them, and know that people are trying to get your attention, get your money, and that if it's truly scientific, just trust me, it's boring, and that's okay. That's sometimes how you know that it is just scientific information. Okay, so let's talk about theories versus laws in terms of science theory, science law. So scientific thinking, like we said, is a systematic approach to understanding the natural world, right? And that involves experimentation and analysis. And so when we get to scientific theory, that means it's a well-substantiated explanation based on lots of repeated experiments. And it oftentimes can explain why things happen based on a wider range of evidence. But theories still remain theoretical, okay? That it's not, you can't say this was proven. You can say this suggests that such and such happens, okay? That's a theory. Laws, scientific law, by definition, is a statement that describes what happens in nature, often expressed as a mathematical formula, and it never attempts to explain why. It predicts what will happen under very specific conditions, right? Like the law of gravity. That happens every single time. There's a mathematical equation for it, and it's under specific conditions. It doesn't say why it happens, okay? So those are the difference between scientific theory and scientific law. So I just wanted to put up this Venn diagram just to kind of show you the difference between laws and theories and kind of where they overlap. So where they overlap is they're both based on evidence. They can both change with new evidence. We've talked about scientific thinking is always changing, but they can't change into one another. So theories rely heavily on inferences, more generalizations. They tend to want to explain how things happened. Laws are basically based on experimental evidence. and they hold for very specific conditions. So they're more descriptions. They're more like what happened, whereas theory is trying to ask how does it happen or why does it happen, okay? So those are the differences between those two. Okay, so that concludes our lecture on scientific thinking, a little bit about, or not a little bit, a lot about the scientific method, scientific experimental design. So best thing to do would be to go back up, look at what the goals were for today, make sure that you can meet all those goals, and then just take some time to sit with it, take notes, ask me any questions if you need to, and that concludes our lecture on scientific thinking.